Okay, so what I want to do today is go over this paper. I'm not going to go over the entire thing. I'm just going to go over the abstract, basically. So it's called Zinc and the Modulation of NRF2 in Human Neuroblastoma Cells. So neuroblastoma cells, these are cancer cells, which are neurons. So neuroblastoma, let's go to Wikipedia. So, neuroblastoma is a type of cancer that forms in certain types of nerve tissue, and it's most frequently starts from one of the adrenal glands. So, remember, these adrenal glands are part of the nervous system, but they produce a lot of very important hormones for your body. So, for instance, the adrenal glands are endocrine glands that produce a variety of hormones, including adrenaline and the steroids, aldosterone and cortisol. So think of the adrenal cortex. This is where you get your corticosteroids from. So neuroblastomas are a type of cancer that are formed most frequently within the adrenal glands. So that is, just so you know, that is what a neuroblastoma is. So, zinc and the modulation of NRF2 in human neuroblastoma cells. And NRF2 is a protein, but it's also a signaling molecule. So we'll go over that. I mean, might as well go over it right now. So NRF2 has another name. So it also goes by nuclear factor erythroid 2 related factor 2, so or NFE2L2. So what we like to do in biochemistry is have a million names for the same freaking thing. So here there are 3, 4 different names for NRF2. Just know that it has multiple names. And it's a transcription factor. So transcription factors are things that will interact with the DNA that make the DNA either express itself or unexpress itself. So transcription factors are factors that change the rate of transcription of certain genes. And so when DNA gets transcribed into RNA, that RNA then gets translated into a protein. So if you alter transcription, you end up altering the concentration of the protein that that gene ends up creating. So there are multiple levels of regulation in the production of proteins, but transcription factors are one of them because they affect transcription. So it is a transcription factor that in humans is encoded by the NFE2L2 gene. NRF2 is a basic leucine zipper. So leucine zippers, just so you know, it is a type of motif that proteins can have, and it's usually the result of a coiled coil. So here we have 2 alpha helices or 2 coils. I shouldn't say it's a coiled coil explicitly. Let's go to it real quick. A basic leucine zipper domain or BZIP domain is found in many DNA binding eukaryotic proteins. So it's a common motif or it's a common domain in many proteins that repeats. It's not necessarily with the same amino acid sequence, but a similar amino acid sequence, or at least similar in that they allow for this shape to happen. So let's see if there's anything else. So one part of the domain contains a region that mediates sequence-specific DNA binding and the leucine zipper. So here you can see these 2 coils are interacting with the DNA molecule, and then the leucine zipper, in between these 2 helices, there are quite a few leucines, and those leucines are both hydrophobic, and they allow these 2 coils to coil together. So knowing that NFE2L2, also known as NRF, has this basic leucine zipper, which allows it to interact with DNA, and that interaction with DNA is how it is a transcription factor. Okay. So, and specifically, this is a transcription factor that regulates the expression of antioxidant proteins. Okay, so you have proteins that have antioxidant capabilities associated with them. So, for instance, one type, I don't know if it's if NRF2 explicitly will upregulate this, but there is catalase and there's superoxide dismutase. Those are two different antioxidant proteins or two antioxidant enzymes that your body produces to help control or at least help remove harmful free radicals and harmful oxidants out of the system. So this NRF2 is one of the proteins that regulates the expression of these oxidant proteins, and it explicitly does it to protect against oxidative damage that is triggered by injury and inflammation. So several drugs that stimulate NRF2, the NRF2 pathway, are being studied for treatment of diseases that are caused by oxidative stress. So let us, knowing that zinc modulates NRF2 in human neuroblastoma cells. So human neuroblastoma cells, cancers, usually have really screwed up expressions of a lot of different proteins. In fact, by definition, they have screwed up expressions of proteins. So some of the important proteins associated for cancer and for survival are the pro and antioxidant hormones, or sorry, the pro and antioxidant enzymes and proteins, because these will play a role in helping the cell decide whether or not it lives or dies. So, continuing, so dopamine, here are the highlights. Dopamine neurotoxicity is exacerbated by zinc deficiency. So when there are high levels, very high levels of dopamine expressed, this can cause neurotoxicity and convince cells to die. So let's see if Wikipedia has neurotoxicity. So I don't think I'll be able to find one real quick, but just know that at high levels of dopamine, that can cause cells to die. So zinc deficient cells show an impaired dopamine neurotoxicity induce HO1 upregulation. So what is HO1? This is heme oxygenase. So this is associated, it's a stress-induced isoform present throughout the body with high concentrations in the spleen, liver, kidneys. HO1 is an enzyme this big that contains 288 amino acid residues. So let's see what it does explicitly. Oh, and that was explicitly, yeah, for heme oxygenase 1. So what do the heme oxygenases are enzymes that catalyze the degradation of heme, and this produces biliverdin, which is one of the things that I think it's bilirubin. So I believe as this biliverdin gets processed all the way down to bilirubin, which I believe gives your feces its color, but don't quote me on that. I would have to look at the mechanism again, or metabolic pathway again. So this heme oxygenase 1 is a specific isoform of the heme oxygenases, and this one is upregulated as a result of stress in the body, and this is encoded by the HMOX1 gene, and the studies found levels of HO1 in lung tissue were directly related to infection with tuberculosis or infection-free areas, and knockout mice were found susceptible, showing an essential role of this enzyme. So it appears as though a study has found the levels of this were related to tuberculosis or infection-free areas, and the knockout mice were found susceptible. So if you remove heme oxygenase abilities from mice, it ends up screwing up their ability to fight against infection. So that's just one of the options, or one of the things that is associated with HO1. And here another thing is it apparently is associated in neuroblastomas as well. So zinc deficient cells have an impaired ability for dopamine neurotoxicity induced HO1 upregulation. So we'll probably read more about this later, but when you have dopamine neurotoxicity or high levels of it, normally you have HO1 upregulation as well. But zinc deficient cells show an inability to upregulate heme oxygenase 1. So that seems to be a problem. So low zinc availability leads to tubulin oxidation. So tubulin is it's the protein, the monomer, if you will, that produces the microtubules in the body. So this is an example. This is the 3D structure of tubulin. And here you can see an example with an individual tubulin protein. And these individual tubulin proteins will bind together to form these tubes, also known as microtubules. And these are extremely important in neurons. In fact, in neurons, I believe these are capped in a way that prevent them from breaking down at all. But normally these are being formed and degraded over time, and this helps transport things across cells. So if there's low zinc availability, it leads to tubulin oxidation, and this oxidation of tubulin decreases or is associated with the decrease in NRF2 nuclear transport. So this NRF2, remember, this is a transcription factor. So for it to work properly, it needs to get into the nucleus where your DNA is at. And if the tubulins are oxidized and there's an inhibition of NRF2 to get into the nucleus, then NRF2 will not be able to do its activity. It will not be able to regulate the expression of, it won't be able to regulate the expression of these antioxidant hormones or antioxidant proteins. So low zinc availability also inhibits the degradation of the NRF2 repressor BOC1. So BOC1 is another protein, and BOC1 is normally degraded. Proteins are normally synthesized and degraded over time. But if you put a cap on the degradation, if you put a cap on the degradation of BOC1, then BOC1 is going to be more and more concentrated within the cell. And so if you increase more and more the concentration of more BOC1, then those BOC1s are going to repress NRF2 from doing its job. So let's do a search Google for BOC1. Okay. Drink. So this is a transcription regulator protein as well. So it is also a transcription factor in some way. So this gene encodes a transcription factor that belongs to the cap and collar type of basic region leucine zipper factor families. So remember, basic region leucine zippers, or BZips, was also what NRF2 was. The encoded protein contains broad complex, this stuff is not too important. So it functions as a repressor of MAF recognition elements, or MARES, M-A-R-E, and transcription is repressed. So these will bind to these MARE elements on DNA, and then it will repress the transcription of that particular gene that is directly downstream of that area. It might, I don't know if it's downstream or upstream, but it will do one of those. It'll repress something. So I think that's pretty much it. Just know that this represses areas that it can bind to. So Bach is going to repress NRF2's ability to NRF2, and I don't know exactly how it represses it. Maybe the paper will go into more detail. So here they're saying zinc is crucial in the neuronal response to dopamine-induced oxidative stress. Okay. So continue. Let's go to the abstract, and we'll just. Oh, perfect. They have a good visual that we can look into. So, abstract. Zinc plays a key role in the modulation of neuronal redox homeostasis. Excuse me. So redox. These are reduction and oxidation. So you can have oxidizers and you can have reducers, and your cells, just by living, will be producing reactive components. So you have redox or reduction oxidation homeostasis that has to be maintained within the cell for the cell to survive properly. And in fact, sometimes the cell can use the dysregulation of redox to initiate apoptosis or cell death. So zinc is playing a key role in the modulation of the neuron's ability to maintain redox homeostasis. So a decreased zinc availability is associated with neuronal NADPH oxidase and nitric oxide synthase activation. So if you don't have enough zinc in the cell, the neuronal NADPH oxidase, and NADPH is just another, how do I phrase that? This is an enzyme that, or not an enzyme, this is a chemical that is used to reduce a different chemical. So this H on NADPH is almost like a negatively charged hydrogen. It functions similarly to a negatively charged hydrogen. And in fact, let's just look this up. So you can have NADP plus, which is the oxidized form, and you can have NADPH, which is the reduced form. And I wish they had a, what's it called? A chemical equation. But basically, you can add this hydrogen, and you can also add, I shouldn't even say it's a hydrogen with a free electron. It's almost like a hydrogen with two free electrons. So basically, you can add, frequently what this is used for is it can take a proton from the water, and then it can take this hydrogen with two electrons associated with it, and then it can use those to add two hydrogens and two electrons to a different chemical. And that is a way to reduce that other chemical. And so you can have a balancing act between NADP plus and NADPH, which just know that those are being used to transport electrons and hydrogens around. So think of it as a hydrogen currency or an electron currency within the cell. So similar to how ATP is an energy currency, NADPH and NAD+, those are electron currencies within the cell. Anyways, zinc, a decrease in zinc availability, this is associated with neuronal NADPH oxidase and nitric oxide synthase activation. So this will activate the oxidase and the NO synthase. And so, how do I phrase this? This is all associated with redox signaling or reduction oxidation signaling. So remember, these are free electrons, and nitric oxide is also a, um, if I believe correctly, I don't remember if it's a radical or not, but it's important in signaling, at least within the body. So this causes a deregulation of the redox signaling and impaired glutathione synthesis, and glutathione is an antioxidant. So the present work tested the hypothesis that zinc is necessary in the neuronal defense response against dopamine-induced oxidative stress. So when you have uh, a lot of dopamine that's being expressed in a certain area of the body or a certain area of the brain. And this is often associated with trauma. So if you get trauma to an area, there might be a dopamine release in that particular area. And I don't know if it's technically a dopamine release or possibly the vesicles that contain the dopamine might actually in of themselves be rupturing. I wouldn't be able to tell you that myself, but this causes a oxidative stress within the cell, and this oxidative stress can convince the cell that it needs to die. And one of the ways that dopamine induces oxidative stress is through heme oxygenase 1 upregulation. So this heme oxygenase will basically be a way of causing this dopamine-induced oxidative stress. So dopamine, it showed higher cytotoxicity when zinc availability was low. So zinc seems to be playing an extremely crucial part in the body's, what would you call this? So there's a lot of components in the cell that are all regulating each other. So if you have, say, a lot of us just have dopamine, and we don't want those low levels of dopamine to just kill our cells, right? So we have regulatory mechanisms to prevent that small amount of dopamine from causing upregulation of heme oxygenase upregulation and thereby preventing our cells from just dying. So how do we have that regulation in place? Well, some of that regulation sometimes requires metal cofactors or other chemicals. And so if someone is deficient in zinc, or at least has low bioavailability of zinc, and one of the things that can cause this is zinc can literally accumulate in certain cells and not become bioavailable, and it can actually basically precipitate out of the solution. Regardless, there are ways to make zinc unavailable in the brain. So dopamine showed higher cytotoxicity when zinc availability was low. So this is telling us that zinc is probably it plays a role in the regulation that prevents this oxidative stress. So humans, IMR32 neuroblastoma cells responded to high dopamine concentrations by upregulating HO1. And this upregulation involved NRF2 translocation to the nucleus. So the NRF2, this nuclear transcription factor, gets transported into the nucleus through those microtubules. And how do I phrase this? This human IMR32 neuroblastoma cells responded to high dopamine concentrations by upregulating heme oxygenase 1, and this upregulation involved these nuclear factors that we were talking about earlier. And these get transported into the nucleus, and degradation of the BOC1 repressor and the NRF2 DNA binding, but it was independent of ERK1/2 activation. Don't worry too much about that. So the dopamine-mediated induction of HO1 expression was dependent on the concentration of zinc in the medium. So these IMR32 cells, which is these cancer cells, in fact, these neuroblastoma cells, which are cultured, if they were incubated in zinc-deficient medium, it showed an impaired response to dopamine with lower heme oxygenase 1 mRNA and protein levels that control dopamine-challenged cells. So this altered heme oxygenase 1 upregulation was reversed by zinc supplementation. Zinc is preventing HO1 from being upregulated. So in the presence of dopamine, this NRF2 nuclear translocation and BAC1 degradation were lower in zinc deficient cells. So in zinc deficient cells, NRF2 was not able to get into the nucleus, and BAC2 wasn't being degraded as readily or as easily as it should be. So the mechanism involved include one, impaired NRF tubulin interaction. So remember, the NRF2 gets translocated into the nucleus through the microtubules, so it needs to interact with tubulin in some way. And so zinc might be playing an important role in allowing the tubulin to bind with this nuclear factor. And alterations in the proteasome mediated degradation of BAC1 secondary to decreased ubiquitination. So ubiquitin is a protein that is used to target some proteins to be destroyed, and so alterations in the proteasome mediated deregulation of BAC1 secondary to a decrease in ubiquitination, excuse me. So the results suggest that zinc is crucial in the neuronal response to dopamine-induced oxidative stress, in part through its role in the modulation of the NRF2 and the BAC1-driven upregulation of heme oxygenase 1 expression. So here we have a graphical representation, and that is too small. Let's go to the high res. Here we go. So dopamine, remember, dopamine is a small chemical, so I believe it can just soak through the cell membrane without an issue. So it gets into the cell membrane, and here it causes NRF2 activation, and then NRF2 gets transported into the cell, or into the nucleus, and this normally would cause heme oxygenase 1 expression. And then, but when you add, and when you have zinc deficiency, this nuclear transport gets downregulated. When you have zinc deficiency, this BAC1, which prevents, and BAC1 apparently is also interacting directly with NRF2. So I might have misspoke saying that it interacts with the DNA, which it probably does that as well, but apparently it's also interacting with NRF2. So BAC2, when there's zinc deficiency, this also gets the degradation gets downregulated. So both of these combined ends up reducing the amount of heme oxygenase 1, and then this causes neuronal death. So there's an upregulation of neuronal death. Now, sometimes you want cells to die, right? Sometimes sometimes the cells are cancerous, sometimes cells have received too much damage, and so it'd be better for those cells to undergo apoptosis, and so perhaps you do want the redox dysregulation to convince the cell to die. That might be something that you want, but you want to reserve that for very specific cases. So you don't want someone to have what is CTE? You don't want people to have chronic traumatic encephalopathy, CTE. I can't, it's all Greek to me. So for instance, in CTE, you get repeated damage to the brain, and that is causing the brain to undergo programmed cell death in a lot of cells, and it's a neurodegenerative disease. So, and the symptoms do not typically begin until years after the injuries and can include behavioral problems. So, that might be an instance where we might want to consider zinc supplementation and chelators that improve the bioavailability of zinc so that these neurons do not convince themselves that they need to die. So granted, this seems to be a more long-term thing. Granted, it's neurodegenerative, so it seems a lot of times when neurons die, the nearby neurons will also might also get the signal to die as well. But I think it's the release of necrosis factors, but don't quote me on that. I'd have to reread the those cellular signaling papers. Regardless, this is what this lab had not uncovered, because we did know already that zinc played an important role in preserving neurons from dying, but here they're elucidating that the mechanism, which this is what biochemists like to do. We like to know the mechanisms of how things work. We don't like to just say this works because we tested and it works. We want to know how things work. We want to know what, basically, we're reverse engineering how biology is maintaining homeostasis. So here, they learned that at least some of the things, at least a few of the things that is preventing the cells from dying is the downregulation of the nuclear transport of NRF2, the downregulation of the degradation of BOC1, and therefore the downregulation of heme oxygenase 1, which is then upregulating neuronal death. So, focus. So with this, so what does this tell me? Well, especially in the context of COVID-19, I think there is a possibility that there might be some dopamine upregulation in corona patients, because dopamine, so how do I phrase this? There is a peptide that is associated with the POMC molecule, the POMC molecule. And this peptide is associated with the suppression of prolactin. This peptide is associated with the suppression of prolactin. And so now I'm concerned that since this amino acid sequence that's at the beginning of the POMC molecule, there is an almost identical sequence of about 14 amino acids that are on the spike protein of SARS-CoV-2. So my concern right now is if we say make antibodies against this particular sequence, and we probably will because it's on the spike protein and it is exposed, and then also the major histocompatibility complex might also just clip it out and present it for the white blood cells to see. So it is possible that we make antibodies against this amino acid sequence. And so then those antibodies will go and attack this amino acid sequence on the POMC molecule, or at least the peptides that are associated with the POMC molecule. And then this would increase prolactin. Yeah, well, actually, I'm rambling. Just know that dopamine also plays an extremely important role in the suppression of prolactin. So, yeah, I'll have to go over this. I'm just worried that this entire system, the dopamine system, uh, as a part of the pituitary adrenal pituitary hypothalamal pituitary adrenal axis, the HPA axis. I'm afraid that all of this is getting screwed up by coronaviruses in general, but I think this SARS-CoV-2 seems to be extremely adept at doing that. So yeah, I think I'm going to have to look over all of this just to make sure I have all the pieces straight in my head, because they're all just sort of tumbling around in there right now. So with that, I'm going to quit, and I'll try to get the other POMC videos up. It's a long review, so it's been taking a while to get those videos done. So with that, thanks for watching, and I will see you tomorrow, hopefully.